Molecular design determines whether the chosen biology can be engaged effectively, safely, and commercially. For decades, medicinal chemistry has been iterative, labor-intensive, and heavily dependent on wet lab experimentation. A compound is synthesized, tested, modified, and retested. Each cycle consumes time, capital, and physical infrastructure. The structural constraint has always been experimentation bandwidth. Deep learning and generative AI change that constraint fundamentally. Deep learning models learn complex relationships between molecular structure and biological activity. Generative AI systems create entirely new molecular structures optimized for predefined properties. This does not eliminate laboratory validation, but it dramatically reduces blind experimental iteration. The strategic shift is from brute force synthesis to probability-guided design. Generative models learn statistical patterns from large chemical datasets. Architectures such as variational autoencoders, generative adversarial networks, and transformer-based molecular models capture the distribution of chemically valid compounds. Once trained, these systems generate novel molecules that satisfy defined constraints such as binding, affinity, solubility, molecular weight, and toxicity thresholds. This is directed generation, not random expansion. Instead of screening millions of molecules without structure, generative AI narrows the search space intelligently. It proposes candidates with higher probability of therapeutic success before synthesis begins. Early discovery cycles compress. From a leadership perspective, the economic implications are substantial. Every unnecessary synthesis avoided reduces cost and shortens timelines. Wet lab experimentation consumes both financial and calendar capital. Reducing dependency on physical iteration increases throughput without proportional infrastructure. Expansion generative design alone, however, is not sufficient. A molecule that binds effectively to a target is not automatically a viable drug. Absorption, distribution, metabolism, excretion, and toxicity, collectively known as ADMET properties, determine long-term viability. Deep learning models trained on historical pharmacokinetic and toxicology datasets can predict ADMET characteristics computationally. These models evaluate molecular structures and estimate solubility, permeability, metabolic stability, off-target effects, and toxicity risk. When generative AI integrates with predictive ADMET modeling, a closed-loop optimization system emerges. The generative engine proposes molecules, predictive models evaluate them across multiple parameters. Iterative refinement continues computationally until candidates satisfy multi-objective constraints. Only then are top-ranked molecules synthesized, laboratory work shifts. From exploration to confirmation, attrition decreases because liabilities are identified earlier. Modification becomes cheaper and more precise. Deep learning excels in modeling nonlinear structure, property relationships, traditional quantitative structure. Activity models rely on handcrafted descriptors. Deep neural networks learn hierarchical representations directly from molecular graphs or string encodings. Graph neural networks treat molecules as interconnected atomic systems, capturing relational structure explicitly. This improves predictive precision and candidate prioritization. Fewer compounds advance into animal studies or early clinical evaluation. With hidden weaknesses, strategic realism remains essential. Generative chemistry performs best when high-quality training data is abundant. In therapeutic areas with extensive historical chemical libraries, models learn meaningful structure activity patterns. In novel biology with sparse data, predictive reliability declines. Data maturity determines model strength. Generative AI is most powerful within well-characterized chemical spaces. Less reliable in unexplored domains. Leadership must evaluate data readiness before committing large-scale generative programs. Synthetic feasibility presents another constraint. A model may generate chemically valid structures that are impractical to manufacture. Integration with AI driven retrosynthesis planning ensures proposed molecules can be produced efficiently. Deep learning-based synthesis prediction evaluates manufacturing feasibility before laboratory commitment. Without this alignment, generative output remains theoretical. Optimization discipline is also critical. If objective functions focus narrowly on binding affinity models, may produce compounds with unacceptable safety or pharmacokinetic profiles. Multi-objective optimization frameworks balance efficacy, safety, and drug-like properties. Leadership oversight defines these priorities. Model interpretability supports adoption. Deep learning systems can achieve strong predictive performance while remaining opaque. In regulated environments, understanding why a molecule receives a favorable prediction strengthens scientific confidence and regulatory communication. Explainability techniques such as attention mapping and feature attribution enhance transparency. Cultural integration matters equally. Medicinal chemists may initially view generative AI as encroachment. Successful implementation positions AI as collaborator rather than replacement. Human expertise remains essential for assessing novelty, plausibility, and strategic direction. AI expands idea generation capacity. Scientists curate, refine, and validate. From a portfolio perspective, generative AI increases exploration breadth within fixed budgets. Instead of investing heavily in physical screening, libraries, organizations explore vast virtual chemical spaces computationally. Innovation probability increases. Time compression strengthens competitive positioning. In competitive therapeutic areas, first mover advantage matters, AI enabled. Design shortens the path from validated target to candidate. Nomination. Earlier nomination extends effective patent duration and accelerates revenue realization. For CDMOs, integration of generative design and process modeling enhances partnership value. Sponsors seek innovation, aligned manufacturing partners. For biotech firms, generative AI reduces initial capital intensity by minimizing early wet lab expansion. Governance discipline remains essential. Models require external validation. Continuous monitoring protects against performance drift. Blind reliance on internal training datasets increases bias risk. Generative AI is not a magic engine. It is a design amplifier operating within statistical boundaries. Leadership confidence must align with methodological rigor. When implemented systematically, deep learning and generative AI shift molecular design from reactive iteration to forward engineered optimization. Instead of synthesizing molecules to discover properties, organizations predict properties to determine which molecules deserve synthesis. This inversion is strategic. Reducing wet lab dependency strengthens scientific focus by concentrating experimentation on high probability hypotheses. The greatest impact is not cost reduction alone, it is probability elevation. Improved candidate quality before preclinical entry reduces downstream attrition. Early ADMET risk detection decreases safety surprises. Faster design cycles increase pipeline throughput. Compounding improvements across multiple programs transforms portfolio economics. Organizations that lead in AI-enabled molecular design will integrate generative systems, predictive, ADMET modeling, synthesis, planning, and human expertise into coherent workflows. Computational creativity becomes therapeutic advancement. Deep learning and generative AI do not eliminate uncertainty, they restructure it. Failure detection moves earlier, iteration cycles compress, exploration bandwidth expands without proportional capital growth. The strategic decision for leadership is clear: continue scaling physical experimentation linearly or augment chemistry with non-linear computational leverage. In a landscape of tightening timelines and growing biological complexity, generative AI offers a mechanism to redesign molecular innovation economics, not as a substitute for chemistry, but as its most capable collaborator. Even incremental probability improvement in this domain creates transformative financial and therapeutic value. This is not optional experimentation. It is strategic acceleration.